Great. Wow. Good evening, everybody. Thank you for coming out to the first of the 2023-2024 MES webinar series. Really appreciative to Roger Rittmaster, who's our vice president, who really took initiative this year to get these planned really well in advance, which just made things run really smoothly, and we were able to get a really great series of speakers for this year. My name is Hillary Peterson. I'm the president of the Maine Entomological Society, and I decided this year to have just a short little intro presentation to MES, since we do have non-MES members that join these webinars. And so Maine Entomological Society, why would anybody be interested in bugs? Thank you to Bob Nelson for making this intro presentation. So first of all, what is MES? We are somewhere around 170 individuals from throughout the state of Maine, and some from away, who are interested in the insects and other terrestrial arthropods, including spiders, mites, ticks, that are found in the state of Maine. We were founded in 1997 at what is now the Vowles Arboretum in Augusta, and 10 of the original 14 founders are still active. And we function as a common clearinghouse for information about Maine's amazing insect fauna. So why does it exist? Most of our members are highly interested amateurs, including numerous Maine master naturalists. We provide a general forum for interaction and an exchange of ideas. We also do have entomological professionals as members too, but most professionals and academics are, because of their work, focused really on specific insect pests. And, you know, quote unquote, pest insects are perhaps at most somewhere like 75 species statewide, whereas we have probably well over 20,000 species. So, you know, MES is not a forum to talk about insect as pests. We're a forum to celebrate the biodiversity of insects. So what do? We publish a quarterly newsletter with news and views on Maine insects. It's called the Maine Entomologist. We also conduct monthly field days from late spring through the early fall and much more. So this is a clip of our table of contents that you can find in our newsletter. And you can see that we announce the different field days that we're going to do. We also have a website. This includes lots of different things. We have an archive of past webinar presentations like these. We have archive of past newsletter articles. We have information on each insect order that's in Maine, and much more. We also have a Facebook page called Maine Insects, and at this time we have over 6,200 followers. So this is more of a forum. It's not everybody in here isn't an MES member, obviously, but people have joined this. It's become pretty popular. It's sort of a ask what the idea is and talk about insects kind of Facebook page, so that's great. And we also have a scholarship fund that can help fund Maine-based insect studies. We're always looking for donations to that scholarship, and we have on the website now a donation button for that. MES sponsored 13 entomological bioblitzes at Acadia National Park over the years, adding many hundreds of species to the known fauna in the park, including numerous first state records. And MES members are part of 4 major faunal inventories in Maine. Members have also spent thousands of hours volunteering at the Maine State Museum. And I'm in this photo, which feels wrong because I think I was there like 2 days that someone came and took pictures for the news. I think Charlene really should be on there more than me. And finally, we just participate in lots of things that pop up. So in 2022, we provided help in a project that resulted in the pink edge sulfur being designated as Maine's official butterfly. And then last but not least, just pointing out, up next, December 14th is going to be Are Insects Declining in Maine by Frank Drummond. So without further ado, I'm going to stop sharing my screen.
Thank you, Hillary. So again, I'm Roger Rittmaster, Vice President of MES and the host of this meeting. It's my pleasure to welcome Monique Raymond. I first met Monique last year at a workshop on insect taxonomy And what I remember most about that is besides Monique's enthusiasm, was the cockroach race that we had. But anyhow, Monique is a research technician now at the University of New Hampshire, a collection of insects. And she's going to be defending her master's thesis in the near future, and hopefully will be doing a Ph.D. after that. So I think we're going to hear a lot more from Monique. In her past, she was also a technician at the Bishop's Museum in Honolulu, one that I got to know back in the 1980s, and was very appreciative of their support for some of the identifying some of the fish that I was photographing when I lived out in the South Pacific. So Monique's going to talk to us today about some of the parasitoids that affect blueberry plants. And Monique, I'm going to turn it over to you if you want to start sharing your screen.
All right, sounds good. All right, that looks okay?
Yeah, it looks great.
Okie doke. All right, so well, thank you, Roger, for the introduction. As you just mentioned, my name is Monique Raymond, master's student at the University of New Hampshire. For 3 more weeks, for 2 weeks and 6 days, I will be defending my thesis. So the end is in sight, and I'm looking forward to what comes next. Today, I'm going to be sharing the story of our process identifying the Blueberry Gall Midge and its parasitoids for biocontrol purposes over the past 2 years. And here's the spin on this, all right? This is how it's a little bit different from one of those typical IPM or biocontrol stories, is both the Blueberry Gall Midge hosts and the specialist parasitoids, they're both considered dark taxa. So Diptera and Hymenoptera are some of the most diverse insect orders in Insecta. And so our ability to accurately identify what we collect is limited. It's limited to the resources that are presently available to us as far as, keys and knowledge and species descriptions. And so the relationships that we report for IPM or for biocontrol purposes, they may be less precise than we actually need them to be if the taxonomic background of these groups is relatively poor. And Cecidomyiidae in particular, this subfamily is known for being very, very diverse, very, very speciose, and there are so few experts in this group. So if you know any promising young dipterists, you should send them in the direction of working in Cecidomyiidae, because Ray Gagne is not going to be around forever, unfortunately. You'd be my first choice of people to make immortal. So as much as this project is an IPM or a biocontrol applied project, it's mostly taxonomic in nature. And this idea of illuminating dark taxa and cryptic organisms by testing and creating new species concepts, that's something I've been fascinated with since my first intro bio class at UNH in 2011, which was taught by Don Chandler, who I'm very fortunate to share an office with today. And that feels a little surreal. I look up to him so much. And the question of how we define a species, that is so exciting. It is the conceptual foundation of our understanding of every living thing on Earth, right? That's a big, heavy, exciting idea. And I think taxonomy is a kind of love letter to the incredible biodiversity that nature has provided us. And entomology in particular, that is a perfect representation of that diversity and the evolutionary processes that bring it about. Anyway, it is so important to note that there are many different kinds of species concepts, and they have changed through time, and they're not all created equal. You can see here on the left side of the screen, there's I think it's a blog, but he lists 26 different types of species concepts, from classical morphological concepts to genetic concepts to behavioral, ecological, chemical, and chromosomal. I mean, you can really break it down into so many different ways of defining a species. But integrating these concepts for a given organism or a group of organisms and testing and creating a stronger understanding, I just think that stuff is really cool. So applying that to agriculture, that's a way to do this like really nerdy, really niche work with, a certain purpose that I think helps food systems and sustainable agriculture. And you can really feel good about, yourself at the end of the day, helping save the world, one tiny species description at a time. So Cecidomyiidae or Cecidomyiidae, these are the perfect group to really break down these species concepts through. So this family, Cecidomyiidae, it's suspected to be the most speciose animal group on Earth. They are known for their adaptive radiation to different host plants at the generic level within plant families. And they're real funky little critters. They're small, and their larvae actually have been observed to experience patogenesis. So the sexually immature larvae have been known to reproduce, which is really strange. And some Cecidomyiidae, they actually present haplodiploidy in some cases. So this is a different manner of reproduction where you can give or take the sexual aspect of it. And this is more seen in Hymenoptera. So to find it in flies, that's really unusual. Altogether, there are about 6,600 global described species of Cecidomyiids, but the estimation in recent years is anywhere from tens of thousands, like 20,000 to 40,000, all the way up to 2 million extant undescribed species. So these critters, they're very good at doing what they do, and we have a lot of work to get names for as many of them as we can. And the reason why this matters, right, is because these are pests of major crops worldwide. The most well-known one would be the Hessian fly, which is a pest of wheat. It's pretty well known in Europe. And then you have the apple and pear leaf curling midges. And of course, relevant to us here in New England, we have the blueberry gall midges. And this is also referred to as blueberry tip midges. So blueberry gall midge and blueberry tip midge, they're both in blueberry. And that's specifically Vaccinium is the genus. And then the section within Vaccinium, it's the Cyanococcus section, because cranberry and blueberry are both Vaccinium. So they just kind of distinguish it with sections. So for our intents and purposes, the blueberry gall midge is 2 species, 2+ species. So the Blueberry Gall Midge Complex, BGMC, this is made up of Dasyinura oxycocana and Protoplosis vaccinii. And Dasyinura oxycocana, it's the most widely reported and studied all the way from across the United States, I mean Maine to Washington, all the way out to Italy and Korea. So this is pretty well known. And the damage that it causes blueberry crops, it's been estimated that the damage could cause anywhere from 20 to 80% crop loss in some cases. But like Hillary mentioned in her introduction here to this presentation tonight, we're not just talking about pests, right? Where this isn't like a pest-oriented story necessarily, just one part of it. So both Dasyinura oxycocana and Protoplosis vaccinii, they are cosmopolitan, found worldwide. Both have been described as univoltine and multivoltine, so having one generation versus multiple generations in an active year or an active season. And so there's conflicting literature. The verdict is not out on their life history in that way. And when they do occur in the same places, they are known to have overlapping phenologies. They are in the same subfamily, so Cecidomyiidae, then Cecidomyiidae, they're both in that group, but they are in separate tribes and separate super tribes. So they have just adapted to infest blueberry plants separate from one another. Neither has actually been reported in New Hampshire. And this is kind of interesting because New Hampshire is actually home to the first blueberry cultivation that we know of. That would be Frederick Coville in the early 20th century. Although I think New Jersey likes to claim being like the blueberry capital of the world. But I mean, like, have you been to Maine at the same time? We're all like fighting for that blueberry capital title. But New Hampshire may be the original home of blueberry cultivation. And Dasyinura oxycocana, by the way, is known to be present in Maine. In fact, Frances Drummond and Judith Collins have done some excellent work in Blueberry Gall Midge in Maine specifically, and that their papers have been really foundational to my research. So just a little life cycle information, the adult blueberry gall midge deposits her eggs in buds of blueberry plants. And so she will either walk into the bud or somehow, I don't know, just maneuver to get inside the bud and lay the eggs in there. And Dasyinura oxycocana is known to infest both floral buds and leaf buds. And so this, in the leaf buds, it's often the apical meristem of the plants, which is kind of home to the stem cell tissue of the plants. It's where that new growth generates from. And this is arguably like a very important plant tissue. It has a huge impact on the health and development of the entire plant. So the majority of the life cycle of the gall midge is spent as larvae. They have 3 instars of larvae inside the buds, and they are eating the plant tissue from the inside out. And while this happens, they're actually deforming the leaves, and it produces this really interesting, like bound, curled, enrolled, tight bundle of leaves. And it always reminds me of like a hand-rolled cigar or cigarette or something like that. You can see in this image on the left, this one's very dried out, but look how tight these layers of leaves are bound to each other. It's actually really tough to separate them out. So the larvae are essentially, they're living inside their food source, and their deformation of the plant tissue provides perfect shelter. Excuse me. After 3 instars, they drop out of the leaf, out of the bud, to the soil, and they pupate in the soil and then emerge as adults, and they will live anywhere from 1 to 3 days or so. And the leaf damage actually looks different depending on if you're looking at high bush or low bush blueberry. And this hasn't been really documented before, just something that I've observed. So here on the left, this is high bush blueberry from New Hampshire, Vaccinium corymbosum, and the damaged leaves, the tips, we usually call them tips, they tend to take more of like a curved, almost a sickle shape or crescent moon shape when they're really bad. Meanwhile, in the low bush blueberry down here on the bottom, Vaccinium angustifolium, the tips will make almost a right angle, and they look like flags or wind socks almost. And it's very distinct, very easy to see. There are also some differences in the like blistered texture of the leaves as well, but that could have to do with like the age of the leaves. So because of this bundled protective nature of the plant tips, the larvae are shielded against many insecticides. So non-systemic insecticides, the ones that are sprayed or applied to the outside of the plant, this would be maybe one of the safest ways to treat these pests, but just they're so deep inside the buds. I mean, good luck finding them, good luck getting to them. So because of their very, very small size, the larvae being less than 1 millimeter and the location in which they develop, it's really hard to detect them and monitor them in order to initiate treatment. And so that initial presence and abundance, it's determined through this painstaking dissection of tips. And normally by the time you see the tips being warped, it's a little too late, right? Because now the next generation is already developing. So one really strong indicator of the blueberry gall midge's presence and abundance is actually the abundance of specialized natural enemies like parasitoid wasps. So here they are, my babies here. So Platygastrid wasps, I think they are so cool. They are very, very small. If you can see the scale bar here, 100 micrometers. Most platygastrids are also less than 1 millimeter in length. And they tend to have this really distinct black, shiny appearance. They tend to have, their head oriented downward and maybe a little bit of a humpback. And they have all these like interesting adaptations to their exoskeleton to accommodate their reproductive structures. So this one that you see here, it has this hump on the underside of its abdomen. That's the ovipositor, the female reproductive structure, curled and coiled inside of that body part. Some folks are familiar with one of the like one of the more well-known genera in Platygastroidea is Inostema. And that is the one who has a horn up on its back. It just kind of goes right from the anterior part of the abdomen on the top dorsally, and it's just this big horn, and the ovipositor just goes all the way up into that horn like a sheath or a sleeve. It's really cool. But altogether, Platygastrids, they are a family of about 2,000 described species, although like Cecidomyiidae, it's anticipated that there are many, many, many more. And these are solitary insects. They are coenbionts. They are egg larval endoparasites. So they deposit their eggs inside eggs or larvae of other species. And we don't know for sure, but we suspect that they are oligophagous. So they tend to prey upon and target a small number, but a multiple number of hosts. The subfamily Platygastrinae is specialized against Cecidomyiidae. So this is really promising. This is really convenient for us. And we know that in the past, that Platygastrids have been successfully used for biocontrol against both the African and the Asian rice gall midges, the Swede midge, apple and pear leaf curling midges, and a new problem that's cropping up in the Midwest right now is the soybean gall midge. It was reported just a short number of years ago, and they just really got to the point where they were able to start identifying parasitoids, because it's getting pretty bad in soy in the Midwest. They just detected it in several more states. Originally, I think it was Minnesota, and now it's like at least 5 or 6 states. So long story short, this group, very cool. One thing that's kind of interesting about this whole story is that there was a study conducted in the early 2000s down in Mississippi at a USDA agricultural research station. And they were rearing parasitoids out of gall midges down there. And 4 out of the 5 were Platygastrids. Unfortunately, the phenology of these animals is not very well known, and it really requires close monitoring at a site-by-site basis to understand like when they will be there or if they will be there. And there are no practical guides to identify them to the generic level. You can't find a key to identify these to the generic level. And if you wanted to identify them to the species level, you would need an expert on your hands, of which there's just a few in the world. All the way in Romania even. So the existing literature wasn't really specific enough to give us an idea whether what we have in New England would be a good solution to the blueberry gall midge that's occurring up here. And that previous literature, the study in Mississippi, it didn't include any study sites in New England or in the Northeast, and it didn't include low bush blueberry plants, which are very different from high bush blueberry plants. Oh, yes, I'm looking at the chat right now. Sorry, I'm just like getting this little window up. Coenbiont. What is that? Right now, I definitely defined it. A coenbiont is Oh, they parasitized earlier host stages, and the hosts continue to develop once the parasitoid has deposited their offspring into the body of the host. So instead of depositing the egg in the larva and the larva just dies immediately, it allows the larva to continue to develop through all of those instar stages, and it allows it to actually begin pupation. So the parasitoid doesn't kill and escape its host until it's in the soil pupating? That's a really good question. That's one of those words that doesn't come up a lot. So the big picture of what we're doing is we're working to identify these specialist parasitoids for biocontrol of the blueberry gall midge in New England. And along the way, we're hoping to determine if the blueberry gall midge is present in New Hampshire, because it's never been reported before. And if it is here, is it the same species that's found in Maine in those previous studies? So what we did is we took a pretty broad look. We have 9 study sites total. There's only a few on this map so far, but these study sites are in Maine, New Hampshire, Florida, and Mississippi, that original one from that early 2000s study. And so we're looking at both high bush, low bush, and velvet leaf blueberry. You might know velvet leaf blueberry just kind of out in the forest or in a barren. It's mixed in with the Angustifolium regular low bush blueberry a lot of the time. It's not as delicious, not as sweet. It's a little more dark and bitter, but it's edible and it's out there, and it's often intermixed with other varieties. We included wild sites. So we've got, state preserve, we've got abandoned farms and wild blueberry barrens. We also have organic farms and experimental farms, like those associated with universities. One with the University of New Hampshire, Maine, and the University of Florida. So just to get an idea of the variety of landscapes that we're looking at here, these are all 9 in this slide. And up top, these are some of our main sites. The one in the top left, that is the Jonesboro Blueberry Hill Farm that's associated with the University of Maine. And seeing that place for the first time, way up in Washington County, it was very cool. I mean, I don't think I've ever seen that much blueberry. It was like a sea of blueberry up there, and like rolling hills of just blueberry as far as the eye could see. And that farm in particular, it's 2 fields, each 30 acres, so totaling 60 acres. And that was really neat to be in the middle of that, like stand in the middle of that field and know that, okay, everything that I'm experiencing at this exact location is associated with blueberry. You don't have that, edge effect as much right there in the middle. And yeah, so you can kind of see a little bit of the other types of plants that were intermixed in some cases. Not all the way bottom, but the second one up on the right-hand side, that's Paynes Prairie State Preserve, which is outside of Gainesville, Florida. And that was a really neat site. It's fire adapted like the barrens that we have up north, but in a different way. The fire, I think, is a little more frequent, and you have all these different tropical species intermixed, all these palms and different pines and things like that. So it's been cool to kind of consider like blueberry is everywhere, and it plays all these different roles in ecosystems with different types of animals and other types of plants. So just a little look at that. So our collection methods. So for our mass collection, specifically for the wasps, we were doing malaise traps, pound-style malaise traps, as you can see on the left. And these are erected at most of our sites from as early as we could get them out there to as late as we could get them out there. And the bottle of ethanol at the very top of the tent-like structure, that would be changed out about bi-weekly, give or take, depending on like the weather and people's availability to go out there. And we also did yellow pan trapping, or yellow bowls, as some people know them. And this was in place of, some folks use the yellow sticky traps, the yellow sticky paper, but that kind of deforms the specimens. It's really hard to get them off in one piece and like in a nice shape to be examined and identified. So we went with the bowls instead. And we tended to do about 200 bowls at a time, where you just kind of lay them out in the field and try to keep them in a straight line. That way you don't accidentally miss one on, like there's one 20 feet away, and then you come back the next day to pick them up and you forget about that one, and you're just like leaving plastic in these places. So it's really important to set them up in a way that you're gonna pick them all up in the same order. So in these bowls, we just use regular tap water and then about 1 drop of biodegradable dish soap to prevent too much surface tension, because otherwise these really microscopic insects will just bounce off the surface of the water. Specifically, it's good to use a non-sudsing soap, so the kind that's made for dishwashers instead of sinks is best. Anyway, so whenever, at least near us, near the Durham campus at University of New Hampshire, we have an experimental blueberry crop right near the campus. And I would do this yellow pan trapping every time that I changed the malaise traps. So that would be about once every 2 weeks. got a whole lot of material. In fact, I haven't finished sorting it yet. It's a lot to go through. Then we get leaves in there. We get all kinds of other bycatch, some really interesting stuff, even like salamanders, unfortunately. And typically during these collecting events, we would also collect some seemingly infested plant tips as well, in the hopes that we could rear out adult parasitoid wasps from any larvae that would be present, but also to kind of get some of the larvae and try to identify them, at least to the generic level, if not the species level, and know exactly which member of BGMC we're dealing with here. So what we tend to do is we would take a photo of the plant tip out in the field, and then we would document which plastic vial or plastic tube that it went in. That way, when we dissected them, we would be able to look at the photo and say like, oh, okay, it looks like this when there's an active infestation at this site. So for morphological methods, specifically for the Blueberry Gall Midge Complex, this has been quite the experience. So we took the specimens out of the plant tissue by a kind of wet shaking method. I always think of it as shake and bake in a way, because you've got this like sloshing shaking motion. So these plastic vials, I think they're like 50 milliliter plastic vials. They've got this screw top, and then it's a hole in the top. And so we fitted a very fine silk screen mesh under the cap, and that was how we stored the tips to allow anyone who was alive to possibly come out of their host. And we would keep the plant tissue like kind of fresh up to a month. At the very latest, you kind of wait up to a month. And so when it was time to dissect this and get the larva out, we would take off the cap and fill it about a third of the way with water. And then instead of the mesh, we would replace it with pieces of like Ziploc baggies to make it watertight. And you would just shake the heck out of that thing, and then pour it in a Petri dish. And typically that would be good enough to tell if there's any midges present. And if you see a few, then you know it's time. Okay, I'm going to take the forceps and really get in there and try to pull out every single midge. When we did that, we would take all of the larvae that were taken out of a given plant tip, put them on a slide in glycerol, and image them. That way we could get an idea of like which instar are they currently in? Are there multiple species present in this tip? Because that's something that's actually highly likely. And for count, and that kind of thing. And so we image those and then separate every individual either onto its own slide for further examination, or it would go in a microvial anticipating DNA extraction. And when we were imaging the individuals, we would do this through bright field microscopy, but also confocal light scanning microscopy, which we're very lucky to have access to here at the University of New Hampshire. And you can see that down in the picture in blue. So what this is you have like 4 or 5 different lasers shooting different wavelengths of light at the subject. And insect tissue actually autofluoresces. So you can distinguish the different type of tissue, whether it's sclerite or connective tissue or fat bodies or resilin. It just kind of lights up like a Christmas tree. So this guy down here in blue, this larvae is just really a lot of resilin rich tissue. So that's why he's lighting up that color. Yeah, so it turns out you really can't identify these things past the generic level. It's really not possible. There are no keys. But there are a few diagnostic features that we worked with, and we were going to give it a try. Let's see. I'm clicking. There we go. So for the morphological methods for the wasps, this was tricky too. So we developed classical or morphological species concepts through repeating these kinds of sorting exercises. So we took our fresh caught specimens, we took specimens from the insect collection at UNH, and then we also looked at previous studies, illustrations of specimens, right, to compare what we found. So we took this kind of integrated approach to developing these morphological species concepts, and we ended up really focusing on one genus in particular, Cenopeus, because that was not only the most abundant one that we caught, but also 2 of the 4 platygastrids in a previous study had been Entomological. So because we have limited time, limited people, in this first year, it's just Entomological year. Next year will be Platygastroid year. So as far as identifying the other platygastrids in the system, because that's also good information, we actually identified all of them to the generic level, which is, kind of tricky for a new master's student. And we did this by creating a Frankenstein key, which is why this little guy is up on the slide. So we took keys to, I guess you would say, taxonomically inferior subfamilies or subfamilies that have been made redundant, and just species descriptions, personal correspondences. There was a website that was someone's passion project where they did a key to a few different genera. So we kind of took all these resources and integrated them with each other, and we got Frankenstein's key to all of the platygastrids occurring in blueberry at these 9 study sites. So it was quite the feat. It was a lot of fun, and that will be published in my thesis in the coming weeks. Okay, so we're not just looking at morphology to identify these insects, we're also taking a molecular approach through DNA barcoding. So we were using the CO1 gene, cytochrome oxidase 1, which is ultra conserved. It's found in all eukaryotes, but it's particularly useful for DNA barcoding in insects and in Hymenoptera in particular. And this has been proven to really work for differentiating things from each other at the species level or population level in some cases. So for barcoding, we did a non-destructive DNA extraction process. That way we could maintain the voucher specimens to look at their anatomy and their morphology later on, right? We don't want to destroy them forever in order to get their DNA. And this is kind of important because they are so small. If we didn't do this intensive non-destructive method with heating and cooling and extra products involved, we would have to sacrifice, a good portion of their bodies in order to get enough DNA. So we use universal primers. This is really not interesting stuff. Universal primers. There are lots of other primers that can be used both for platygastrids and Cecidomyiidae. We just kind of looked at what was recommended, and these ones seem to work. And then when it came down to the Sanger sequencing, we actually had that done out of house at Yale at their Keck sequencing facility. And let me tell you, I am not going to be a molecular biologist when I grow up. This process has been it's been a rocky road. I mean, it hasn't been awful, but it's been pretty tough. And I will say my biggest grievance with molecular lab work is the lack of natural sunlight in those labs. I mean, it's dark and the light is so bright overhead and it's not ergonomic in there. I just love a comfy, cozy entomological collection with old furniture in there. Oh, I've got a question here. How do you do a non-destructive process? So really the difference between this non-destructive extraction process and a more typical process, the typical one, people are often grinding up insect bodies or even multiple insect bodies and kind of taking all of that mass and extracting the DNA out of it. And then really what you have left over, the specimen that you used, is really kind of broken and not in one cohesive form anymore. So the non-destructive DNA process, when you apply the first, say proteinase K, for example, we actually use double the recommended amount. And we had this kind of cyclical experience of warming it in a water bath. In fact, we warmed those specimens in their vials, the specimen in the vial with the proteinase K, we warm them overnight, as long as 24 hours, and then put them in the deep freezer, and then thawed them, and then cooled them down to room temperature. And so this kind of heating and cooling, it causes the contraction and the expansion of the exoskeleton, and it allows that proteinase K to get inside the body and do what it does inside without having to open up the body segments. So with enough time, with enough heating and cooling, you manage to make it so the fat, the muscle, the soft tissues inside the body, they are completely liquefied, and you can take out like the complete body capsule, including hairs and everything. And you can dry that and pin that and keep it for external examination. So it is painstaking, but it's worth the effort. So when we got our chromatograms back from the Sanger sequencing from Yale, we would analyze them along with reference sequences taken off of these big open source databases. So the Barcode of Life database, which you can see here, bold systems in the top left, they have this awesome public data portal where you have this search bar and you type in, say, Dasyner oxycocona, and it shows you all of these specimen records that have barcoding sequences associated with them. And you can say select all and export, and you get the FASTA files of all their nucleotides in that section, and you can just download it and put it in with your specimens and do, like we did, a maximum likelihood analysis to get a phylogenetic tree, kind of inferring the relationships between these specimens or between their sequences, really. And it's just one gene, so it's only so telling. But like I said, CO1, it seems to work. It's kind of the unanimously agreed upon go-to barcoding gene method. So, I mean, getting the relationships between the specimens, that's definitely helpful, but there are also species delimitation or species partitioning models that are available. There's all these different methods. And the ones that I use to see, you know, is this a distinct species from this, are called ASAP and MPTP. That's multi-rate Poisson tree process. And so I would run different maximum likelihood constraints. In fact, I trimmed the sequences. You can see this like broad rainbow colored array of reads here, and we would trim it. So we would trim off the right-hand tail one time and run it, trim off the left-hand tail one time and run it, trim off both and run it, and see what the differences are. And depending on, you know, the best tree we got, the most well-supported tree, we would apply that to both ASAP and MPTP and kind of see what the differences are. It says, okay, is this 9 species or is this 10 species, you know, and try to come to a consensus there. So we did that for the midges and the wasps, actually, although we had many, many less midges. So the results of our midges here. So we have really limited success in morphological identification. I mean, we spotted some protoplosis. So this one in the upper right-hand corner, this is certainly protoplosis. It might be protoplosis vaccinii, hard to tell, but it has these 2 corniform papillary sensilla in the terminal segment and the bomb end of the thing. And this is distinct to either protoplosis or contrarinaria. So knowing that, you know, it might be protoplosis being in blueberry. Yeah, so we were able to figure this one out. But overall, the diagnostic characteristics that we were really looking for and focusing on, it's not what we saw. I mean, these things, they had all kinds of different papilla and numbers and different seta in different locations. And what Ray Gagne had detailed in all of his wonderful books and catalogs, it was, I mean, they really are so diverse. There's so many different presentations of these characteristics. So like I said, we weren't really able to really identify many of them, most of them at all. So as far as field observations though, it was interesting, the field observations. Some of them were different than what we expected. We did not find any midges in floral buds in any New England sites. But we did observe that there were multiple generations, certainly, in low bush and high bush blueberry. So as far as univoltinism, might not be the case, at least not where we observed. And I have a feeling that the generations of the midges, I think that it might actually correspond with the vegetative flushes that the plants undergo, where they have like new growth from the apical meristem at different cycles. And sometimes they'll rotate between producing new vegetative tissue and then producing new root tissue. So they kind of go back and forth a certain number of times, depending on the species and depending on the location, the weather, and all of that. We didn't find any blueberry gall midge in natural landscapes. So any site that had blueberry but wasn't a farm, we didn't find any in smaller crops. It was really only the larger ones that were pretty expansive, and the plants were kind of well connected to each other. And in the midges themselves, I did not see any differences in color or their mobility, like how they move around and how fast they are and how active they are. And that was just based on personal correspondence from the author of the previous study. So we didn't see any of that, which was interesting. But the barcoding results, right? So these are our molecular species concepts or genetic species concepts. So like I said, we didn't do a ton of barcoding with the midges, but what we got was really interesting. So you can see that our fresh specimens are highlighted in yellow. And the reference specimens that I took out of the bold database, I took everything that was Dasyura oxycocana, and then there was nothing in there for Protoplosis vaccinii. So instead, I downloaded Protoplosis longivula, which is like a close cousin. So I figured, yeah, why not? We'll see what the relationships look like. We'll see if they cluster together. And the Maine low bush blueberry collected specimens, they were all with Dasyura oxycocana. And we knew that Dasyura was occurring in low bush blueberry in Maine. So great, this is what we expected. You know, you love when things work out that way. And then in New Hampshire, the high bush blueberry collected specimens in New Hampshire, they were clustered with that Protoplosis. So I think possibly Protoplosis vaccinii is in this high bush in New Hampshire, Dasyura oxycocana in low bush in Maine. And, you know, maybe there is no connection. Maybe they, I mean, most likely they were not distributed through natural means from one crop to the other. A lot of these infestations are the result of people moving plants around. In fact, that's how they got to Korea. They were able to trace the origins of the gall midge in Korea to like Georgia, New Jersey, and Michigan. So these are definitely separate genera, likely from separate origins. So within these clades, like let's zoom in on the Dasyura oxycocana, that upper cluster there. So all of the red and the single tips, these represent molecular species concepts through these delimitation models. So you can see I labeled them A through E, and based on this model, these 5 are distinct species from one another. And what's kind of crazy about this is that all of these specimens were actually collected at the same place on the same day. And it was that Blueberry Hill Farm up in Jonesboro, blueberry as far as the eye can see. Maybe they were collected from different corners of the fields, or field 1 versus field 2. We didn't record that. But I think it's really interesting that there is this well-supported divergence in these lineages. You can see the bootstrap values, the bigger numbers on there, that represents the level of support for that like junction or splitting of the branches of the tree. And you know, it looks pretty good. It looks pretty solid. We got that 100 up there. So something is definitely going on here. They are not all necessarily having gene flow between each other. They're not necessarily all reproducing amongst each other. There's some kind of isolation, genetic isolation taking place within this field at this exact moment in time. And when I talk about the sequences that were in bold, the Dasyura oxycocana reference sequences, so bold has this algorithm in their own data set, in their own database, that delimits species their own way. And nobody really knows how that algorithm works. It's a proprietary kind of thing. And they call their species level groups bins, or barcode index numbers. And something that's kind of interesting is that for all the Dasyura oxycocana in bold, there were 2 bins. So things identified as this one species are in these 2 big species level groups. And so this 100% split we have in the all the way left part of this section of the tree, perhaps this represents the 2 different bins. Who knows? I mean, maybe we do have 5 species, cryptic species, or a cryptic species complex taking place in this field. But then again, you know, the ASAP model, the MPTP model, both of them together, that doesn't necessarily mean that these, you know, that these are different species. I mean, we have to integrate lots of different methods to define what a species is. And I have theories about this, but I could go on and on and on. So to the wasps again, going back and forth, back and forth. So we had rearing success, and this was like a very exciting victory. It really gained a lot of momentum early on in the project. This was one of our first collecting events where we took the plant tissue, and I think it was within like 3 weeks we observed this adult Platygastrid who was with the little pupal case and everything. It was perfect in there. And it was identified both morphologically and molecularly as Cenopaeus pennsylvanicum. And that is actually consistent with one of the morphological species concepts in this previous study, Sampson et al. 2006. They only had line drawings of their specimens, and so you can see that here on the left. It's pretty close to the one here on the right. And we were also able to match ours morphologically to the holotype, the type specimen of Cenopaeus pennsylvanicum. So this is good news, but abundances really matter. All right, so who was most abundant? 2 Cenopaeus seeming species were the most abundant ones. This is great. So on the top, we've got what we call morphospecies A. On the bottom, morphospecies B. Interestingly enough, they were most abundant at these 2 main sites. So A was in the New Hampshire high bush with Protoplosis, and B was in the Maine low bush with Dasypnera oxycocona. And they seem to be quite consistent with that 2006 study. You know, we didn't focus too much on phenology. It wasn't really built into the scope of the experiments, but I did map the abundances of these Cenopaeus in these 2 main sites, Woodman Farm in New Hampshire and Blueberry Hill Farm up in Maine. And I have more samples to process. We have more data to incorporate in this, and I'd like to start collecting a little bit earlier, because look at this huge jump in abundance at the second seeming generation of Blueberry Gall Midge here. Kind of interesting stuff. So whether it is Pennsylvanicum or morphospecies A or morphospecies B, they tend to follow somewhat similar patterns in abundances and maybe generations that probably have to do with the generations of midges. So the next thing we're going to do, we're going to incorporate more data into this analysis, and we're going to estimate parasitization rates as well. You can see here on the right, this is a DNA extracted gall midge. So this guy's very tiny, and its fat, its muscles have been digested. It's kind of like a ghost of sorts here. And then down in the bottom, this is actually the rear part of the body, that kind of darker brown, that is a parasitoid egg. That is somewhere, probably it was deposited in the gut. So by extracting the DNA and then applying a little bit of potassium hydroxide for a couple of minutes, it's clear as day. You can count the number of eggs, which I think is really cool. So back to these guys, you'll remember I said seemed consistent with the previous study. Well, it turns out that our morphospecies A is not actually what had been, defined in the other study as Cenopaeus viripes. So we looked at the description, we looked at illustrations, we looked at photographs, and what we had at our main high bush site, it was not viripes, and we don't know what it is. In fact, it might be a new species. So for now, we're calling it Cenopaeus near viripes. And yeah, it's kind of exciting. They were, the second most abundant specimens across all these 9 sites. And then we've got, morphospecies B here, Cenopaeus pennsylvanicum, the one that was reared and the one that was most abundant. And yeah, this is actually the most frequent species level identification in all of Cenopaeus. So we've got 400 species in Cenopaeus, and the vast majority of any Cenopaeus anyone finds are put into this group. And so that tells us that the morphological and maybe molecular species concepts are just too loose, too broad. Because when you go into bold and you kind of look up Cenopaeus pennsylvanicum, down here on the left, you can see for 391 specimen sequences that are given this ID, they fit into 4 bins. So hypothetically, 4 species concepts. Well, what we found, we had some somewhat monophyletic groups here. They weren't well supported, not as well supported as the midges, but you can see Cenopaeus pennsylvanicum, that's the big one right there in the middle. Reference sequences, and then our sequences are in bold. They're all intermixed with each other. All 4 bins plus are in there. And look at those red groups and single tips. They're implying through these delimitation methods that this is a cryptic species complex as well, possibly. And then we have our morphospecies complex A, Cenopaeus neoviripes, down on the bottom, well supported, real exciting stuff. And then the Cenopaeus viripes from previous literature, that's all the way up here at the top. So it's clearly a separate group. So I went and looked for the voucher specimens from that previous study because we only had line drawing illustrations. I was actually at the Smithsonian yesterday. It was a great experience. I went through the collection, all these drawers and these cabinets, looking for this collecting event, these few specimens. And the good news is that I found one. The bad news is that this specimen, reared in the same collecting experiment, it was not included in the paper. It's a totally separate species. I mean, you look at it, it doesn't fit into either of these groups. So it wasn't reported. So, I mean, that's kind of interesting. And we found them up north too, but it had never been reported. So they certainly parasitize Diptera oxycocana, which is interesting. So in conclusion, right, what did we learn? Well, the New England BGMC, Blueberry Gall Midge Complex, it might be composed of 2 cryptic species complexes, the Diptera oxycocana cluster and then the Protoplosis cluster. And for our parasitoids, the most abundant and specialized ones, they might be cryptic species complexes as well. And our morphospecies A, neoviripes, that could very well be undescribed. So we're working on describing that. And there is this additional Cenopaeus that was in the Smithsonian that was reared and never reported. So the story continues. There's lots more work to do. I'm going to tackle another genus, the second most abundance genus in the year ahead. And yeah, I would say where IPM research meets dark taxa, the solutions are not always clear, but we have an ethical obligation, right, as scientists and as researchers, to have a solid conceptual foundation for these species or for these groups when we are giving suggestions to farmers and people in sustainable agriculture. I'd like to acknowledge the source of our funding, which is a USDA Farm Bill through the Plant Protection Act, and my mentors, collaborators, including Phil Fanning up in Maine, and the members of my thesis committee. I'd like to invite anyone who is interested to attend my thesis defense on Zoom later this month. And yeah, if anyone has any other questions, I see a question here about what percentage of Blueberry Gall Midge specimens in your research were found to be parasitized by any parasitoids? Good question. So the image that I included of the midge with the egg in it, I actually didn't get like the final puzzle piece of putting them in potassium hydroxide to really clarify stuff until 2 weeks ago. So I've been painstakingly looking at these DNA extracted specimens, and they look like ghosts, like, you know, trying to find anything. And I couldn't find any eggs. I was like, what do they look like? What should I be looking for? And yeah, it wasn't until 2 weeks ago. So as far as, you know, identifying the other parasitoids, we'll see. Some folks say that there's an Aprostocitus, which is a chalcidoid wasp that also parasitizes them. But the platygastrids are specifically evolved in conjunction to Cecidomyiidae. They're much more specialized, more targeted to gall midges. And in that previous study, the platygastrids parasitize the gall midges at about 5 times higher of a rate than the chalcidoid had. So that one is more of a generalist. It's not as effective. And then why is the blueberry gall midge only found in large fields? You know, that's a really good question. I think part of it might be that these larger crops, these are places that are more serious about production. The smaller crops that we went to were kind of like, you know, hobby farms and historic farms, and they're sometimes just one element of a larger agricultural experimental area. So the introduction of new plants in order to increase the area of the crop, I think that is how they got introduced in the first place.
Monique,
Talking
let me
about-
ask you to,
Yeah.
Monique,
Yeah.
unmute, or rather unshare your screen now, and we can have a conversation among all the people here.
Yes. On it.
And thank you
There
so
we
much
go.
for a wonderful presentation. Yes, so we have one more question that you can read in the chat
Yeah.
box, and then if anybody wants to unmute themselves and ask a question, please do.
Sounds good. So the remaining question, can you talk more about the larval forms of the Cecidomyiidae reproducing? Yeah. So thank you, River. So that phenomenon is referred to as petogenesis, and it is very obscure, and it was observed in Cecidomyiids. I think it was just one time. I don't think the observation was replicated at all, but it was observed in one of the more basal lineages in Cecidomyiidae. And those ones, they tend to be mycophagous, not phytophagous. So they're adapted to living in and consuming fungal tissues. So real weird. I mean, don't even get me started about how much we don't know about fungi, right? Because that's a whole different situation.
Are there any other questions? And while we're waiting, Monique, I've got a really dumb question that what's a dark taxon? I keep thinking of the dark web.
Oh, yeah. So a dark taxon, the way that I kind of describe them in a really casual, offhand way is these groups, I mean, they don't have to be in insects. They could be in any living organism. And these groups are often very diverse. So something about their life history means that they are reproducing at a higher rate, or they're introducing more opportunities for adaptation and evolution, like somehow in their life history. And so you've got this tree of life that just exploded. And some of these groups of things, they're just not, perhaps they're not popular, perhaps they're hard to observe. I mean, the gall midges and the wasps, they're both teeny tiny. A lot of folks, they go their whole life without realizing that these are in their lawn, in their bushes. They're all around us all the time. They're just kind of hard to observe, maybe because of their size, maybe because of their behavior. And so because of that, because of their obscurity, there really is a lack of understanding about them. And so you have all these species and you just have a drop in the bucket as far as how many have been formally described and formally treated taxonomically. And I used to talk about this in Hawaii all the time because tropical rainforests are some of the most biodiverse habitats on Earth. And you think about it and you're in a forest. I mean, it could be a tropical rainforest, it could be our forests up here in the north. And you kind of look up into the trees and you look up at the canopy of the trees and you think, there's just got to be so much up there. It's got to be teeming with life. But who is going to be in the crown of a 200-foot tall tree, like assessing all the life forms that are up there, right? So, because of their location, because of their small size, their behavior, because they are incredibly diverse as well, we have, a flashlight in a pitch black warehouse. And so, we're working on describing more. We're working on these huge biodiversity surveys. Like Canada and Germany have put tons of money into biodiversity research. And so we're working at kind of getting an idea as to how many there are. It takes a long time to describe new species, but we're getting a feel for how many. And oh my gosh, it's more than we could even fathom like 10, 20 years ago. It's really cool.
Thank you.
Yeah.
Any other questions? Well, you're a rock star presenter.
I had a quick question,
Yeah.
Yeah.
Go ahead.
Monique. First of all, I really enjoyed that. That was fabulous. What did you said you just put tap water and a little bit of non-sudsy soap, but like one little drop? That's it in the collecting bowl?
Yeah. And it's really kind of funny. Sometimes collecting insects can be so cheap and casual, and we think of like biodiversity researchers and zoologists going on these field excursions. They've got all this cool gear, all this equipment, and they're gonna, figure out exactly what species are out here. And it, that movie Field of Dreams, where you say, if you build it, they will come. Just put a bowl of water down, and the critters show up. It's like magic, honestly. And one thing to keep in mind, I mean, you can do this in your backyard. A great place to do it is alongside streams or in taller grasses. But you want to make sure that you don't fill the water too much, because if it rains, it can overflow and everybody pours out. And you don't want it too low because it can evaporate out. And wildlife have been known to drink the water as well. So even like in the African savanna, like elephants will like suck up the water. So yeah, it's so cheap, it's so easy, and you get to look at the stuff that you would never notice otherwise.
What was your filtering method to filter all the water out?
Yeah, so we took embroidery hoops, they're usually about like, appetizer plate to a dinner plate sized embroidery hoops, and fitted them with silk screen mesh, like as fine of a mesh as you can get. And you just kind of depress it a little bit in the middle, and then pour the bowls with the bugs and with the soapy water. You just pour it right over that. And then when you've got all the bowls emptied onto the mesh, you put it under cold water, just fresh water running from the sink, and just kind of work to rinse any soap off of the specimens. Just there's no more bubbles, no more suds at all. And then deposit that right in ethanol. Yeah, so it's pretty easy. I mean, when I go all the way up to Washington County, I often go camping up there, so I don't have access to like lab facilities necessarily, and I just use the spigot outside some building, and yeah, it's pretty easy. Okay, so colleagues in other countries trying to figure out vaccinium midges and parasitoids, or are we alone? Well, yeah, that's a good question. So since I'm in this weird transition in life between a master's research project and a PhD research project, I'm actually working at finding people who are involved in this. So down at the University of Florida, there is a fruit and vegetable IPM lab that's been doing a little bit of work with the Blueberry Gall Midge down there, but they were focusing on a different group of parasitoids and not really taking as much of a taxonomic approach. And now that young lady, she just got her PhD and she's working with the USDA out in Puerto Rico. So I think she's kind of pivoted into other work, which is great for her. It's an awesome gig. But yeah, the group is getting smaller and smaller, but I'm hoping in the future to be able to kind of carry this into a PhD scale research project in some way. There's a few, there's folks in Romania, there's folks in the United States and Canada. Yeah, and that's kind of it, at least for platygastrids right now. But more, and what's kind of cool is that this group has more women represented in the list of taxonomic specialists than many other parasitoid wasp groups. So bringing feminism into tiny wasps.
Well, thank you so much. Oh, go ahead. Yep. Thank you. This was a great presentation. Really enjoyed it. I know it was a lot of effort on your part. So thank you.
Thank you. Thank you for letting me be the first in your series. It's such an honor.
Thanks so much, Monique. And I put it in the chat, but just email me that information and I'll put it in the weekly forwarding.
Yes, definitely.